Let me tell you about the birds and the bees and the flowers and the trees, the moon up above, and a thing called love. Let me tell you about the stars in the sky, the girl and the guy, and the way that you kiss on a night like this. When I look into your big brown eyes, it's very plain to see that it's time. Okay, so thank you, Dean, for bringing us in with that intro. This video is about meiosis, which is necessary for sexual reproduction, which is necessary to have life on this planet for many organisms. There's also asexual reproduction, but that's for another time. Okay, so bird and the bees, flower and the trees. Why does this happen? Okay, pollen has baby sperm in it. That's what that is. And I'm going to zoom in. There we go. And that is what we call a haploid cell. Now, I should have looked up how many chromosomes are in a typical flower, but it's not typical. They all kind of vary. So we're going to say the number is 8. Okay, so this is the pollen from a flower that got picked up by the bee. And inside, there are 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. Okay, then the bee buzzes over to a flower and drops off the pollen, which swims into a pollen tube and eventually fuses with a haploid egg. Haploid meaning half, right? Okay, so pollen with sperm and then egg, right? So collectively, these are called gametes. You got the male gamete and the female gamete, okay? Gametes. Cool. So here's how you often see that in textbooks. N fused together, sexual reproduction, to make a 2N diploid individual. And oftentimes you'll see this in a cycle, right? A cycle. It's a cycle of life because this diploid individual undergoes mitosis to get bigger and bigger, multicellular organism. I made a human. Maybe it's a plant or it's a piece of fungus, who knows? And then this human undergoes meiosis, which we're talking about today, to produce either sperm or egg. Okay, so this is where our lesson picks up. How does a diploid individual, meaning 2 sets, undergo meiosis to make the haploid, sperm or egg? So I'm just going to do human since I made a stick figure. So we have 46 chromosomes, and then after meiosis is done, we make a sperm with 23 or an egg with 23. And that's what we're going to look at. How does this happen? Lots of videos on meiosis. You got your textbook on meiosis. I'm going to just walk you through it. After I do this with you, you should be able to do this with a friend. Here's what you need. A piece of paper and 4 different colored pencils. So grab that. It could be like a pen, a pencil, and 2 different colored pencils, highlighters. I don't care. It does not matter. But you'll see why. Okay, so we are going to take this and we're going to fold this. Yeah, let's fold this. So 1, 2, yeah, that should work. Okay, so we're going to make, so the paper has 4 columns, 1, 2, 3, 4. And I'm going to hope to fit meiosis 1 on the front and then meiosis 2 on the back. Maybe not. Maybe this way. Yeah, let's do this way. Yeah, okay, let's do it this way. So we're going to have yeah. Okay, interphase. Then we're going to meiosis 1 meiosis 2, and then gametogenesis. That's a big fancy word. Okay, so this is kind of small. I'm going to zoom, but let's go ahead and set up the paper. So this one, this first row will be interphase. So go ahead and label that interphase, and we'll get all some really important terminology down in interphase. Oops, sorry, I'm going further away and I want to go. Okay, so column 1 is interphase. Very similar to mitosis, but also different. All right, moving on. So now we have, we'll call this meiosis 1. And this is really important. Well, all of it is, what am I saying? Next one, meiosis 2. And then I'll just call it gamete formation. It probably has a fancier name, but good enough, gamete formation. Okay, so let's do this, and I hope it works out. If not, you'll still get the gist. Let's start with interphase. So you're familiar with interphase. You're familiar with the whole G1, S, G2, just like mitosis. And that's pretty similar. So not a lot of difference. Just make sure we get the correct whole chromatin, chromosome, sister chromatids thing down. So here's my cell, and I'm going to make a ginormous nucleus so we can see what's going on in this cell. All right, so I'm going to make the cell in G1 because I think it's important. So this is G1, and it is a diploid cell, and we're going to start with 4 chromosomes. 4. So this is why it's important to understand. So this is a chromosome. So 1, 2, whoops, that's weird, 3, 4. So this is my complete diploid organism. It got 2 of these chromosomes from the sperm and 2 from the egg. Okay, so that's what it looks like at G1 when it's doing its thing. Now this cell happens to be in the gonads. It's not a skin cell. It's not an intestine cell. This is a very specific cell whose destiny in life is to become a sperm or an egg. So this is in your ovaries, your testes, and the gamete archeogona and antheridium of the flower. So this is really just specialized tissue to do this process. So that's a key difference, right? All right, so now very similar to what you've seen before, we're going to move on to the S phase. And that is, you probably guessed it, DNA synthesis. So all these chromosomes are going to make copies of themselves. They're still going to be called chromosomes, though, even though they've made a copy. So this is a chromosome. Chromosome. And guess what? When this makes a copy of itself, it's still called a chromosome. Yes, it looks different. And yes, it is different because it is a chromosome consisting of 2 sister chromatids, right? So this, we got to make sure we get the vocab. So 2 sister chromatids. So we've already studied DNA replication. If you haven't, make sure you do. This is identical to this, thanks to the process of DNA replication. All right, nice. Let's just go ahead and make the other chromosomes. Oh, this is a bad pencil. Maybe I'm just too rough. Okay, so there we go, DNA. So this is how we typically see it. Honestly, this is really what we can't really see. Chromosomes normally loose and called chromatin. It condenses, it replicates, and now we have the chromosomes in S phase. Okay, and again, keep in mind what's holding them together is called a centromere. And then, of course, you have kinetochores on there, and the kinetochores fibers, blah, 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 blah. Okay, but we're really just focusing on chromosome number because we're trying to get haploid. So quickly moving down to meiosis. So this cell is now going to enter prophase, metaphase, anaphase, telophase. However, because it's meiosis 1, it's going to undergo prophase 1, metaphase 1, anaphase 1, telophase 1. Okay, so here is the key event, honestly, is prophase 1. This is really important compared to what you saw earlier with mitosis. So I'm going to emphasize it. Sorry, I kind of drew that line. So in prophase 1, okay, the nucleus goes away, so I'll make it dashed. But what you have is homologous pairs of chromosomes. So these are the same homo, right? Same size, same shape, same genetic information, chromosomes. They pair up, okay? So I'm just going to say that like the red has this is homologous with the brown, right? So mom gave the red, dad gave the brown, and they're the same size, same shape, same genetic information. And then I'll make blue. Say the sperm had the blue, and then the egg had the green, and now they're finding each other. And that never happened in mitosis. So this is really important because we're trying to make sure that the resulting haploids have one of each set of chromosome. One number 1, one number 2, one number 3, one number 4. So this is how they do it. Okay, so then after that, it's pretty much what we saw before. Metaphase means middle. So we have metaphase 1, they line up in the middle. And then we have anaphase 1, they pull apart. And then we have telophase 1, they split. Telophase. Okay, so I'm going to do that and then probably like make the video go really fast. So metaphase line up in the middle. So I will have the red and the brown line up in the middle. And then I will have the blue and the red, no, green Hartford line up in the middle. And you got all the same players, you got your spindle fibers coming down, only this time they're coming down to connect with the homologous pairs. And sometimes, just so you guys know, this homologous pair is referred to as a tetrad. Tetra, 4, because there's 4 sister chromatids, right? So, all right, and then of course in anaphase, they split apart. I'm sorry, this will get a little off from my labeling, but that's okay. You get the idea. But this is really important. Anaphase 1 is really important. So the red go to one side and the brown go to the other side. So you can see now I have actually split. I've actually split the tetrad so that I have a nice even separation of genetic material. Okay, boom, 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 boom. And then, of course, it's going to split down the middle here. And so this, entering meiosis 2, we have 2 cells. Okay, and this is key. So at this point, I have the red that ended up in this cell with the green Christmas cell. Okay, and you see that, right? So this is this top cell here becomes this one. And then the bottom one split, and it ended up, because of the way it got pulled apart, with blue and the brown. So, good, I have, even sets. The question is this considered diploid or haploid? What do you think? So the cells entering meiosis 2, diploid or haploid? So keep in mind, we started out with a diploid number of 4, and now we have a haploid number of 2, right? And yes, I know that they're replicated, but it's still 2 chromosomes in each of these cells. So now what we have to do in meiosis 2 is simply get the sister chromatids apart. Okay, so we're already at the haploid stage, but we have the copy that we made. So, of course, you're going to do what any normal cell would do. Maybe prophase 2, we'll just throw it in there. But there's already like no nucleus and stuff. So prophase 2, metaphase 2, anaphase 2, sorry about my handwriting, telophase 2, and of course cytokinesis. Yeah, and this looks just like mitosis. So if you think about it, the chromosomes will line up. That's metaphase too, right? I'm trying to make these a little bigger. Sorry, I'm running out of space. So I'm going to line these up. I'm going to line up the brown and the blue. So they're lined up in the middle, however they line up. Usual players, microtubules, spindle fibers come down, but now their job is not to separate the homologous pairs, but to separate the sister chromatids, right? So then, of course, anaphase happens where they get pulled apart. So yeah, I'll show it. Okay, so my brown got pulled apart. My blue gets pulled apart. And you guys should be doing this with me. My red gets pulled apart. And my green gets pulled apart. And of course, the spindle fibers are doing that. So here we go. And then telophase, you can see right here, it's going to now pinch up through the middle here and down through the middle here so that you result in 4 haploid cells. So this one splits, 1, 2, this one splits, 3, 4, and of course, whatever genetic material they end up with, that's what they end up with. So I got a brown, and I got a blue, and a brown, whoops, throw my pencil around, and a blue, and a red, and a green. Green, green. And then, you get the nucleus that comes back. And then if they're supposed to become sperm, they grow tails and they become sperm. And if they're supposed to be eggs, then one becomes an egg and the rest become what's called polar bodies. So that's kind of weird. But so only one becomes the egg, and the rest kind of dissolve to nourish that egg. But if it's meant to be sperm, they all just grow tails and become sperm.